So, like we just said, heterozygotes are not, heterozygotes are not true breeding. So see if you can figure that one out. Think about it. I'm going to ask you when we go back to class. So, because sometimes dominant traits cover recessive traits, we can't always tell an organism's genes just by looking at it. So we have two different ways to distinguish an organism. We have phenotype and genotype. Phenotype is the physical characteristic that we're looking at. So if we're looking at flowers, there's two phenotypes, purple and white. This is different than genotypes, which is the actual genetic makeup. So it's pretty easy to remember. Phenotypes is physical, genotypes is genes. So if you have purple flowers, that's your phenotype. You are purple, but you could have two possible genotypes, big P, or big P, little p. So that's why it's important to distinguish between the two. So this guy right here, his genotype is big P. He is homozygous. He has two copies of the same dominant allele, so we call him homozygous dominant. His genotype is big P. His phenotype is purple. These two guys, their phenotype is also purple, but their genotype is big P, little p, so they're both heterozygous. This white guy down here, his phenotype is white, but his genotype is little p, so he is homozygous recessive. And you can see that the ratios are important also. So remember that Mendel saw ratios of 3:1 for dominant to recessive phenotype. And that's what we're usually going to see. But that's different than the genotypic ratio, which here is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. So the genotypic and the phenotypic ratios are not always going to be the same. So it's really important to know how can we determine what a dominant phenotype's genotype is. Right? If you have a purple flower, are you ever going to know if it's purple or if it's big P, or little p? How could you know? Right? So the best way to do this is to perform a test cross. And what this means is taking your unknown, your purple flower that you don't know what it is, and crossing it with a true breeding homozygous recessive individual. So you would take your purple flower that you don't know if it's homozygous dominant or heterozygous and cross it with a white flower. Because you know that person's genotype, that flower's genotype, it's going to be little p. And so you can tell by the offspring what the genotype of the parent is. So here's our unknown guy, our unknown purple. Is it big P or big P, little p? We don't know. We're crossing it with a white, and we know this is little p. So when you cross them, we'll get two possible outcomes. Either all of your offspring will be purple, or half of them will be purple and half of them will be white. If they're all purple, you know that you had a big P parent, or it's likely that you had a big P parent. If you have if you get some white offspring and some purple offspring, then you had a big P, little p parent. Because there's no way you could get white offspring unless there was unless you get a recessive trait from both parents. So there must have been a recessive trait in this parent. So that's really important. Now I told you Mendel had two laws. The first one was the law of segregation, which is when the alleles separate during meiosis. So the two alleles that your parents have are not going to be necessarily what you got. And the second one is independent assortment. We talked about this in meiosis, so you should be a little bit familiar with this. Okay, what this is talking about is when we look at one trait like we did right here, this is called a monohybrid cross. Remember, mono means one, and we're looking at crossing two different kinds of things, purple and white, so it's a hybrid. So this is a monohybrid cross. We can do dihybrid crosses when we look at two traits together. So for example, right here, we're looking at two different traits, not just one. This seed is yellow and this seed is green, but this seed is also round and the seed is wrinkled. So we're looking at two different traits instead of just one. This is a dihybrid cross. Okay? So read in your textbook about how to complete that. So looking at a dihybrid cross, Mendel was able to determine the law of independent assortment. This is really important. The law of independent assortment says that the alleles segregate independently of each other. So just because you are yellow and round does not mean that all of your gametes are going to be yellow and round, okay? Your yellowness and your roundness segregate independently of each other. So this is where we get genetic variation from, like we talked about with meiosis. Now, this only applies to genes that are on different chromosomes. So Mendel was really, really, really lucky that all of his 7 traits were on different chromosomes, so it was never affected. Because think of it this way. You know that the genes are on the chromosomes, so if we're separating the chromosomes, if you're yellow on the top of this chromosome and round on the top of this chromosome, then every single gamete is going to be round and yellow. If if they're on the same chromosome. But if they're not on the same chromosome, then they're always going to assort independently. And that is it for 14.1. So 14.2 talks about now probability. So since Mendel figured out all these rules about the number of offspring that were going to be found of each genotype or each phenotype, we can use these to predict now that we know that there's a pattern, which is really great. And so we know these rules because of the laws of segregation and independent assortment. So the fact that the alleles get separated during gamete formation from the parents, and the fact that they all assort independently of each other allows us to know the rules of probability. So your book is going to constantly refer back to tossing a coin because it's the most simple probability problem. So basically, you flip a coin, half the time it's going to come up heads, half the time it's going to come up tails, without fail. Always, if you have a regular coin. And the tosses do not depend on each other. If you flip a coin and it's heads, that's great. The next time, you still have a 50-50 chance of getting heads, every single time. And so, it's the same way with alleles. So if I segregate my two alleles and I say maternal one going this way, paternal one going this way, that does not affect how the next one segregates. Okay? So we have all 23 homologous pairs in a line. They assort completely independently of each other, just like the coins flip completely independently of each other. So there's two rules that we're going to learn about: the multiplication rule and the addition rule. So the multiplication rule is when you're talking about two or more independent events that are going to occur together. So they're all going to occur. So we can look at a monohybrid cross like this. So in a monohybrid cross, what's that say? Segregation in a heterozygote plant. Oh, it went away. Oh, it went away again. Okay. I don't know what that says. Segregation in a heterozygous plant is like tossing a coin. Each gamete has a half chance of giving the dominant allele and a half chance of getting the recessive allele. Every single time. So this is a great little diagram that shows tossing a coin just like the sperm and egg cells during fertilization. So if you are heterozygous, if both parents are heterozygous, so the mom is heterozygous and the dad is heterozygous, then each of the parents can either make a dominant gamete, right here a dominant egg or a dominant sperm, or a recessive egg or a recessive sperm every single time. Okay? And so how the egg gets decided is completely independent of how the sperm gets decided. So it's like flipping a coin with the egg, whether or not you're going to get a dominant or recessive, and it's completely independent, but the same, of the sperm. Okay? So half chance you're going to get big R, half chance you're going to get little r. Same thing up here. Half chance you're going to get big R, half chance you're going to get little r. So the multiplication rule says that you multiply these if the events are happening together. So what are the chances that you're going to get big R, big R? Well, it's a half chance you're going to get up here, a half chance you're going to get up here. So the chance together, you multiply them together. Half times half is 1/4. So 1/4 of the offspring are going to have big R, big R. What are the chances you're going to get little r, little r? Well, there's a half chance you're going to get little r from your mom, half chance you're going to get little r from your dad. So the chances are half times half, which is 1/4. Okay? Now, that is how, and same thing here. What are the chances you're going to get big R from your dad and little r from your mom? Half times half, 1/4. Over here, what are the chances you're going to get little r from your dad and big R from your mom? Half times half equals 1/4. Always multiplication rule. Now, the addition rule comes into play when any one of two or more exclusive events that will occur is calculated by adding together their individual properties. That's a little confusing. Basically, if you're going to be big R, little r, you could either be big R from dad, little r from mom, or little r from dad, big R from mom. It doesn't matter. It's not going to affect you. So you could have this quarter chance or this quarter chance. So overall, your chance of being heterozygous is half. So the rule of addition can be used to figure out that the probability of an F2 plant from a monohybrid cross will be heterozygous rather than homozygous. So we can use, and so if we're looking at a monohybrid, it's not really that exciting that we can figure out this stuff using probability because it's pretty easy to figure out on your own. There's only 4 choices, right? There's 4 boxes. You're going to be one of the 4. It's pretty easy to add up. But it gets really confusing as we get to di, tri, quad, hybrid crosses. It gets crazy. So the ability to use math to help us is going to save everyone tons of time by drawing Punnett squares. So that's what we use it for. So basically, if we're looking at a dihybrid cross, that means 2, right? So we're looking at 2 characters. Like we're going to look at seed color and seed shape at the same time. They're basically 2 monohybrid crosses just happening at the same time. So it's really easy to just use math to figure it out. And so remember, if 2 events are happening at the same time, we use the multiplication rule. So the probability, so we're flashing back to, sorry, I'm skipping back in here. Flashing back to this cross right here. So one parent is big Y, little r, and another parent is little y, little r, little r. So remember that we're looking at those 2 traits together. Y is seed color, R is seed shape. Big Y is yellow, little y is round. I'm sorry, big Y is yellow, little y is green, big R is round, little r is wrinkled. Okay, so that's what we're talking about. So that slide go. So the probability of ending up big Y is the probability of ending up, the probability of ending up homozygous dominant for both, big Y, big R, is just the probability of ending up big Y, which we know is 1/4, times the probability of ending up big R, which is also 1/4, so it's just 1/16. So that's really easy. Then if we're going to figure out the probability of ending up big Y, little y, and big R, it's exactly the same thing, except the numbers are a little bit different. So the probability of ending up heterozygous for seed color is 1/2, just like the probability of ending up heterozygous for anything, times the probability of ending up homozygous dominant for seed shape. So 1/2 times 1/4 is 1/8. So 1/8 of the offspring will be big Y, little y, big R. So what this picture comes from in your textbook is it says, what fraction of the offspring, from a different cross. Okay, so this cross, this is on page 270 in your textbook. It's looking at 3 traits. So this is a tri-hybrid cross. So this is going to save us so much time and so many giant Punnett squares. Okay, this would be 8 squares by 8 squares. That's how big the square would be. So this is much easier than drawing that Punnett square. Think of all the mistakes you can make there. So there's 3 traits, and we're looking at crossing something that's big P, big Y, big R, by big P, little y, little r. So those are our 2 offspring. I wish I could write that on here for you, but I can't. It's on page 270 in your textbook. On the right-hand side, the second paragraph from the bottom. And so it says, what fraction of the offspring from this cross would be predicted to exhibit the recessive phenotypes for at least 2 of the 3 characteristics? So which offspring are going to be recessive for 2 out of the 3 characteristics that we're looking at? So the way to do this is to just write down all of the possible genotypes that would give that to you. So right here we have recessive for the first 2, dominant for the third one. Recessive for the first and last, dominant for the middle. Recessive for the last 2, dominant for the first. And then completely dominant for the first one. Where's my mouse? Recessive for the second, and all recessive. Okay, because remember, big P is the only one that we had a parent that had 2 possibilities to give that to us. So you write down all of those, and then you just figure out all the possibilities. So the probability of getting little p is 1/4. Probability of getting little y is 1/2. Probability of getting big R is 1/2. Multiply them all together. Events happening at the same time. Multiplication rule. 1/4 times 1/2 times 1/2 equals 1/16. You do that for all of these, and then you add them up. Because these ones are not, these cannot happen at the same time. You can't be big P, little y, big R, and homozygous recessive for all of them. You can't do that. So that's why we add them up at the end, and we get 6 out of 16, or 3/8. So 3/8 is the probability that you'll be recessive for at least 2 out of the 3 genes. No, that's crazy. Read it in your textbook and try and work it out to see if you can get the same answer.